Much better job, and I'll keep it brief. So Jamie did her PhD at James Cook University in Australia, and after that she did some conservation work in Tasmania, and I was lucky enough to lure her back to the States a few years ago, and she's been working with me as a postdoc the last few years. She's leaving, literally packing up her car and leaving tonight for a long field trip in Panama, and then to start her faculty position at New Mexico Tech. So look for great things from the Voyles lab. And I promised Jamie that I would not gush too much, and she said that it was okay if I said one nice thing. So I will just say that Jamie is the best, and I hope you enjoy her talk.
Great. Thank you to both Jim and Bree for that overly kind introduction. Yes, let's see. Can we just, check the notes? And just check, can you hear me in the back? Okay. Yes, okay, excellent. So thank you so much for having me here today. It's really my honor to talk to you here in the MVZ. I've been here at UC Berkeley for a little less than a year, and in that time the MVZ community has been so welcoming and just had a huge impact on me, opened a lot of doors personally and professionally, and I really appreciate the opportunity to talk here today, so thank you for coming. Now, since this is my last day at UC Berkeley, and because these MVZ lunches can be a bit informal and fun, I'm going to do something slightly unconventional and present my research in sort of a story format for today. So it is a story that many MVZers are familiar with because it concerns amphibians and the loss of amphibians around the world. And it's a story that is ongoing. So this story is slightly unusual because it is part murder mystery and also part choose your own adventure, right? Because the end has not been written yet. But like all stories, it has some very essential elements. We have a setting that takes place in far off exotic lands. We have some very colorful characters, including the human research characters. And there's an element of suspense, and hopefully I will leave you today with a message of hope. So I'm going to begin this story following that prologue. Here we go. Once upon a time, in the late Devonian period, on the planet Earth, wondrous beasts appeared among the Sarcopterygian fishes. These beasts diversified and multiplied and filled the earth and stayed for over millions and millions of years. They saw great extinction events, and they suffered through interminable ice ages. They saw many other species come and go, and they are still here today. We know them as amphibians. Now, amphibians that we have today on planet Earth are incredibly diverse in form and function, in life history and ecological characteristics. And in my time here at UC Berkeley, we celebrated the description of over 7,000 amphibian species. And I feel very lucky that I was here at the time that Dave Waken and the AmphibiaWeb team were describing and presenting these the 7,000th species. I think we're at 7,040? 40. Which is fantastic. In that short time, species have been described from Papua New Guinea, from Brazil, from northern Pakistan, from India, and even from New York City. So it's quite incredible. Just as we are describing these incredible species, we are simultaneously documenting the complete and total disappearance and loss of other species here on the planet, which is particularly devastating, especially for those of us that really appreciate and enjoy researching these fantastic animals. Now, amphibian declines were first described at the World Congress of Herpetology in 1989. This was the first World Congress, and you might notice that there are some particularly eminent scholars among them. I think I've identified some of the most important. You have Marty Crump. I think this is Hobart Smith and Linda Trube here, and of course, no, not.
That's Roger.
I was hoping you guys could help me with this. Okay. We're going to have to scan the whole crowd a bit more closely. Could someone please tell me who this character is? Because I think I identified Jim McGuire, but I'm not completely sure.
It's grouchy like me.
Well, this grouchy character, who's not Jim
high school
McGuire.
then, right, Kim?
Brad, I thought I had you pegged. Well, this grouchy character, who's not Jim McGuire, was part of this funny bunch that basically was responsible for making the first anecdotal observations about amphibian declines. Now, at first it was really difficult to determine if disappearances were due to true declines or if they were due to natural population fluctuations. But evidence of true declines continued to mount, especially because a lot of these disappearances were happening in very pristine locations, at the very tip-top of mountain peaks, and also in protected national parks. So when you have dramatic declines like this, with no clear indication for what's going on, it's only natural to start hypothesizing about what the causes could be. Now, there are a lot of ideas that were floated around to explain this phenomenon. Now, there's absolutely no doubt that habitat destruction is contributing in a significant way to amphibian declines. We're also losing amphibians due to overexploitation in the form of harvest for food. Amphibians are competing with invasive species, and we also have the hypotheses that UVB radiation and climate change are contributing to their demise. But the problem with all of these causes is that they don't explain why amphibians would disappear from pristine locations and so very rapidly. Which brings me to infectious disease. And the disease that I work on is known as Chytridiomycosis, and it is caused by the fungal pathogen Chytridium gingivalis. And for the rest of the talk today, I will just refer to this pathogen as BD. Now, what you see on the right-hand side of the screen is a composite image of a corroboree frog, and it's compiled from hundreds of electron micrographs. If you zoom in on the surface of the frog, you will find BD in the most superficial layers of skin. This is a pathogen that was first described in 1998. And at the time, there was a lot of skepticism that this could be the cause of amphibian declines for a couple of reasons. First, fungal pathogens aren't known to be particularly lethal without predisposing conditions. Second, this is a pathogen that was only found in the top layers of skin, so it seemed really unlikely that this would cause death. And the third reason is that traditionally we don't consider infectious pathogens to be a realistic driver of permanent declines or extinction events. So there was, as I was saying, a lot of skepticism, and this sets sort of a murder mystery story in place. So I want to set the stage, and I want to give you some information about particular places where we saw these declines. Now, this is a phenomenon that is occurring all over the world. In this map, what you see is a color code for areas where there have been very few to very extreme number of amphibians that are listed as threatened, endangered, or extinct. In these areas where you see the warmer colors, oranges and reds, we're talking about anywhere from 50 to 100% of the amphibian species being listed in these threatened categories. Now, I want to focus in on two very specific areas. One is right here close to UC Berkeley, the Sierra Nevada mountains of California, and the other area is the Cordillera Central of Central America in the Republic of Panama. Now, to start with, I'm going to take you down to the faraway and exotic land of Panama. The Cordillera Central is a mountain range that runs from east to west in Panama. I'm going to put a map up here to show where this mountain range spans, just along here. Now, amphibian declines were noticed in Costa Rica in the 1980s, and they continued until they were just near the border in the early 1990s. Now, with the projection that amphibian declines would continue into Panama, researchers set up study sites all along the Cordillera Central. I was very lucky to work in this site known as El Cope, which was a spectacular place to be a young researcher studying frogs. El Cope has an incredible amphibian assemblage, very high species richness. Amphibian abundance is also very high, as you can see here where I'm surrounded by these beautiful lemur frogs. Now my objectives in El Cope were twofold. I was interested in profiling amphibian immunity, and we were also screening for disease in these amphibian populations. Now, screening for disease began back in 1998, and it continued well up, all the way up until 2004, when BD was first detected in a dead frog that was collected from the side of the stream. Now, there was a subsequent mass mortality event that we documented in great detail. We were able to collect lots and lots of dead animals from the sides of the streams and clearly link the appearance of the pathogen with the mass mortality events that happened in this area. So ultimately what this study did was to show that this pathogen can spread into an amphibian assemblage extremely rapidly. It can cause mass mortality events that are very dramatic, and it can kill amphibian species across a very wide range of phylogenetically distant amphibian taxa. So this is a true murder mystery, but we're zeroing in a little bit on this pathogen. I also want to bring you back closer to home to the Sierra Nevada mountains of California. They're very different from the mountains in Panama, not only because they are in a different climatic region, but they also have a much higher altitudinal range. But the amphibian declines are no less devastating. And in this area, we have a smaller species assemblage in terms of number of species. And this species of frog is one in particular that has been very devastated by BD. Now, Vance Friedenberg and colleagues, who was here at NVZ, with the advent of improved diagnostic techniques, was able to monitor the spread of BD at a much finer regional scale. So in this map image, what you see is one basin that contains lots of lakes, which are these irregular shapes. Now, I'm going to just scan through some images over time, and what I would like you to notice is the colors of the lakes change. They should go from uninfected frog populations that then are detected to be positive for infection, and then the black color indicates lakes where frogs have essentially had an outbreak and a mortality event. So in 2004, everything looked relatively healthy. In 2005, we have more disease spreading through the basin. 2006, we've lost several of the lakes, several of the frog populations in these lakes. And by 2008, you can see that the majority of the basin has lost their frog populations to this disease. Now, what this looks like in real life, you see a picture here of one of these lakes in the basin. And today there's very, very little amphibian life. I snapped this picture of a dead metamorph just a couple weeks ago. There's very few individuals, and they are still dying in these basins because of the spread of the disease in this region. Okay, so now I've set the stage in two very different locations. I'd like to introduce the characters of the story. So for today, the villain, as you would probably guess, is BD, our pathogen. The victim is the host, the amphibians, and I also want to introduce this character, which is the environment. In this story, the environment is the conspirator. And the reason we have these three elements, or these three characters, is because disease is really and truly a dynamic interaction that depends on all of these characters. So disease development completely depends on the host and on the pathogen and on their shared environment. So to begin with, I'm going to tell you about the villain, BD. bee's life cycle begins with a single infectious zoospore. This is an aquatic fungus, as I mentioned, and these zoospores are able to swim by using their flagella, which you see just at the back there. This zoospore will encyst and develop rhizoids for nutrient acquisition. Now, as this develops further, it becomes what we call a sporangia. And in mature sporangia, the cytoplasm will cleave and form new zoospores on the inside. Eventually, an ejection tube will develop, and these zoospores are released out into the external environment. It's at that stage that these zoospores can infect an amphibian host, and specifically, they can colonize frog skin. So I'd like to give you a look at these guys. What you see in culture here is zoospores that are swimming. We have colonies of sporangia on either side and one in the middle here. Now, if you look close in this mature sporangia, you can see zoospores that are swimming within there before they are released into the external environment. So those are the basics of BD. Next, I'm going to talk about the host, the amphibian victims in this story. As I mentioned, the zoospores do infect amphibian skin. Now, at this point, the zoospores can be transmitted either to another adult amphibian or to amphibian tadpoles. So they can be infected at 2 different life stages. Now, amphibian skin is a very important physiological organ. It's responsible for regulation of the exchange of respiratory gases, so oxygen and carbon dioxide. It maintains osmotic balance, which means the ratio of water to electrolyte concentrations, and it's involved in amphibian immunity. So based on some very preliminary findings back in my early days, we found that blood results indicated very low electrolyte levels, and we made this hypothesis. We suggested that BD may disrupt a cutaneous osmoregulatory function, leading to osmotic imbalance and death. So this was the idea that should have connected the villain to the victim and explain how our victims were dying as a result of infection. But how do we test it? Well, to start out with, we monitored for clinical signs of disease in a series of infection trials. This that you see right here is a healthy green tree frog. And healthy green tree frogs can maintain normal posture. They also have a very clear and white, in this case, ventral epidermis. Healthy amphibians normally molt, so they will shed their skin, and normally it comes off in one big entire piece, and then they normally eat it, which is very lovely for this beast that we have. So that is what happens in a normal healthy amphibian. You will find entire sloughs of skin from the whole body. If we look at the histological level, what you'll see in a healthy amphibian is normal layers of epidermis and dermis. Now, in contrast, in a diseased amphibian, what you see is that they cannot maintain normal posture. They're extremely lethargic and unable to move very well to avoid capture. Additionally, what you see in the ventral epidermis is a lot of redness and sort of irritated looking skin. Their skin sloughs come off in irregular, tattered pieces. And if you look at the histological level, there are a lot of pathological abnormalities in the epidermal and the dermal layers. So in order to tie these clinical signs in with a quantitative measure, we use quantitative PCR, and we have tracked the development or the buildup of pathogen load in the skin by swabbing the frog skins at various intervals after inoculating the treatment groups. So this is time since inoculation, and on the y-axis we have BD equivalents, or the intensity of infection, how much BD is in the skin. And what you'll see is that frogs that ultimately become diseased and die have a significantly higher amount of pathogen in their skin. So BD load increases in diseased amphibians. Now this is a problem for amphibians because, as I mentioned, their skin is a physiologically active organ. So the internal environment has to remain hyperosmotic relative to the outside environment, which is hypoosmotic. hypoosmotic. In addition to that, they also have to take in electrolytes such as sodium and chloride. Now the way that they do this is by using specialized cells. So in this little zoomed in portion right here, we see these skin cells separating the external environment from the internal environment. And if you think back to some of your basic cellular mechanisms, we know that these electrolytes, sodium and chloride, which are charged ions, move across skin cells by a process of passive diffusion and ATP-mediated active transport. Now, water flow occurs when you have an exchange of these charged particles across the surface of the skin. You can measure this process using techniques in electrophysiology by placing probes on either side of the skin and measuring the change in the current and the voltage across the skin. Now that's just what we did for a series of experiments. I'm only going to show a couple of graphs. Basically, all I want to demonstrate here is that the electrophysiology in normal healthy amphibians is very different from what you see in diseased amphibians. So what this clearly demonstrates is that we have a dramatic change in the electrolyte transport properties in amphibians that become diseased. In addition to those measurements, we took a lot of samples from the blood and the urine and looked at all of these variables, which are markers for organ functioning and general health. The most consistent and the most robust results come from the blood electrolyte concentrations. They are significantly decreased in amphibians that become diseased. Particularly in potassium and sodium, over the course of infection, we see a drop in these electrolyte levels. So electrolyte concentrations are low in these diseased frogs, and you might not think that's terribly important, except that these electrolytes are really critical for one very big and important organ, and that is the heart. So amphibian heart, all hearts depend on very tightly regulated ratios of electrolytes, and potassium in particular. Especially in frogs, this really depends on having proper concentrations of these electrolytes in circulation. So in these frogs, a healthy normal frog will have electrical events that time the contraction of the heart. Over 20 seconds, this is what the heart rate is in a normal frog. Now if we slow down one of those electrical events over the course of 1 second, this is what that electrical event looks like. As frogs get closer and closer to death, and this happens over the course of a few hours, you get a dramatic change, not only in the heart rate, but also in the electrical characteristics of this signal. So it becomes increasingly disorganized, and the amplitude in the signal is much lower. So ultimately what this results in is asystolic cardiac arrest and mortality. So just to recap all of that information, if the question is how does BD kill frogs, it's really a cascade of physiological events. So it begins with BD load increasing in the skin, there's disruption to skin functioning, a loss of electrolytes from circulation, and ultimately cardiac arrest. So this series of experiments essentially provided an important key to this mystery, this murder mystery, and that is the smoking gun. So now we can conclusively connect the direct cause and effect between our villain, the pathogen, and our victim, which is the amphibian host. So that is not to say, I just want to give a caveat in there, that all species suffer the same fate. There is, or I should say, many amphibians do have very sophisticated immune systems. Okay, there are both innate and acquired components of their immune systems. This is just one part of the story in terms of amphibian defenses, and it's part of a project that I worked on when I was in Panama, in El Cope. So each of these species has a different chemical composition of antimicrobial peptides in their skin. When those peptides are secreted at the skin surface, their job is to fight pathogen invasion and also to ward off predators. So it's a defensive mechanism. Now the chemical signatures from all of these species are different. So here's a wide range of variation in these chemical signatures, and I've highlighted the colors that match each of these species. Now, if you take these secretions and you put them in challenge assays, you can test for inhibition of BD growth, which would presumably happen at the skin surface when BD is infecting these frogs. The results show that these species have very different effectiveness in terms of inhibiting BD growth in vitro. So up here, this species should have strong resistance, is what we predicted, based on their ability to inhibit BD growth. This species had moderate effectiveness at inhibiting BD growth. And down here, we predicted that these Atelopus species were not going to do so well if they became infected with BD. Now, currently, we know that this species is still found in BD-infected regions. This species has been mostly extirpated from BD-infected regions. And there are a lot of species that we just don't have enough information about. But please keep this in mind, because I'm going to come back to this slide in a little while towards the end of the talk. So, with that caveat, and with the introduction to these characters, I'm going to move on from the victim and tell you a bit about the conspirator, which is the environment. So in the BD life cycle, this whole process will vary in terms of the rate of BD growth depending on temperature. BD has a temperature range that goes from about 4 to 27 degrees based on early experiments. So looking at this graph over here, what you see is temperature on the x-axis, and on the y-axis you're looking at BD growth according to optical density measurements with mass spec. So essentially what we had thought was that there was an optimal temperature around 21 degrees for BD growth. And presumably around 4 degrees, BD could not grow as effectively. Now for part of my PhD, I developed methods to look at the BD turnover rate after one cycle of this BD's life cycle. So I was interested in this subsequent generation of zoospores and how many were produced and how long they were active. So these were the three measurements I was looking at: timing of zoospore release, maximum zoospore densities, and how long those zoospores were active in culture. Now, I was particularly interested in understanding what the environment might be doing to this process over multiple generations. Experimental evolution is a technique that's used a lot to investigate adaptive shifts in other pathogens, such as viruses and bacteria. But it has not really been applied to fungal pathogens to the same extent. Now the technique here is to use serial transfer experiments where you propagate the pathogen in culture and control for all variables except the ones that you want to manipulate. For this series of experiments, I was interested in temperature. So I took one BD isolate and propagated it in two different thermal ranges, one at the optimum and then at the suboptimum of 4 degrees, which was the low temperature condition. Now at the end of this propagation, I performed a reciprocal transplant experiment and just swapped the thermal conditions for these lineages of BD. Now I'm just going to show the results in sort of step-by-step fashion. The first that I want to show here is at the optimal temperature. At 23 degrees, what do we see? What we see is that zoospore production peaks and then drops down, so the full life cycle is completed in just under 10 days. In contrast, when you look at BD in 4 degrees, it has a dramatic change. There's two things I want to point out here. The first is that there's a much lower maximum zoospore density in the lower temperature. But there's a longer period of zoospore activity, dramatically longer, to the tune of weeks. So the trade-off for VD in these two thermal conditions is that we are trading maximum fecundity for the long-term growth rate of the pathogen in these two thermal conditions. Now this is for the lineage that was maintained at optimal temperatures. What I'm going to do is overlay the results from the temperature that, or excuse me, from the lineage that was maintained in low temperature conditions. Okay, so in the blue, what you will see is a similar result in terms of maximum zoospore density. At 4 degrees, there is a much lower number of zoospores in the subsequent generation. But also we have a much longer period of zoospore activity. And what you may notice here is that in both thermal conditions, we have a shift in the lineages that were maintained at the lower temperatures. So taking all of these empirical data, we parameterized a mathematical model to estimate population growth rate. And we did find that, in fact, these lineages are behaving very differently. And what this suggests is that there are complex shifts that happen in response to temperature conditions. So why is this important, or why would this matter at all for the pathogen? Well, if you remember these two different exotic places where we have outbreaks happening, the temperature conditions are incredibly different. Remember that Sierra Nevada mountains are basically the frogs in the Sierra Nevada mountains have their body temperatures just above freezing for much of the year. So thinking about that and thinking about how the pathogen is also experiencing these different thermal conditions, I thought a lot about this ideal growth pattern that was established for VD in the very early days. And this is my hypothesis that I'm still working on with a couple of students here at UC Berkeley. My thought is that isolates from temperature regions should grow across a wider range of temperatures than isolates from tropical conditions. And I think that because the frogs and the pathogen is going to be having fluctuations not only seasonally, but also daily, because they are occurring at such high elevations. So the test for this is pretty straightforward. We take isolates from these two different climactic areas and test for zoospore production in different temperature conditions. So across this range, from 0 up to 30. And this is very, very preliminary, but what we have found with the help of two students, Tom Porton and Rochelle Kelly, is that if we plot maximum growth, the profile for temperate isolates is very different than it is for tropical isolates. So not only is the temperate isolate performing better at the higher end of the thermal envelope, but also at the lower end, and we don't think we've yet established the true thermal minimum for these temperate isolates. So again, this is something that is ongoing, and I look forward to presenting more information about this. But so far, comparisons of temperate and tropical isolates are suggesting that there may be some interesting pattern there to explain differences in isolates between these between these climactic regions. Okay, so we have a whole lot of information about these characters. We have a lot of information about the setting of this story. Let's get to the heart of it all. We do have disease resulting because of these characters, and the question is, where is this going? How does this story end? So to put this in a visual map, the question that I have is, down here in Panama, after we have this incredibly devastating, catastrophic, mass mortality event, what happens next? What's going on there now? And the idea that I want to bring out here and highlight today is this concept of epidemic fadeout. Epidemic fadeout is the transition away from the outbreak period of a disease to the enzootic state, where mortality may still happen, but the level of disease is much lower than it is at the epidemic outbreak. Now, this has been discussed for a long time, and it's thought that there are two mechanisms that can lead to epidemic fadeout. The first is when pathogens exhaust their susceptible host species, it leads to extinction. Now, this actually is thought to be biologically improbable, and that's because as host densities are reduced, the potential for transmission is also reduced. So it's thought that this could not occur, but in a recent paper by DeCastro and Volker, they went through all of the empirical studies and outlined 3 potential mechanisms by which disease-induced extinction could occur. One of the mechanisms is that you may have small host populations with very restricted ranges. If you also have rapid pathogen transmission that is not host density dependent, that can also lead to extinction. In this scenario, you have a pathogen that is maybe frequency dependent, which requires contact of hosts that will happen regardless or irrespective of host densities. And the third is that pathogen persistence can occur outside of the host. Now, there's a lot of evidence to suggest that these 2 mechanisms are happening with Chytridiomycosis, and this third one is still being nutted out. So the jury is still out on that one. In any case, we do have some evidence that extinction as a result of this disease has happened. These 2 species are the poster children of the amphibian declines phenomenon. This is the gastric brooding frog, which occurred in Australia along the eastern edge of Australia. And this is the golden toad of Costa Rica. Both of these species had very restricted ranges. They were last seen in the 1980s, and they are thought to be extinct. In fact, I would probably be so bold as to say they are extinct today. I think a lot of people would agree with that at the moment. Now, the role of Chytridiomycosis in their extinction is equivocal. We haven't definitively linked BD to the extinction of these species. So there may have been other factors involved. But if it turns out that Chytridiomycosis was involved in this, this provides, without a doubt, the best empirical evidence of disease-induced extinction, which is a whole new thing that has not been thought to be possible before. Okay. The other mechanism that can lead to epidemic fade-out is when you have rapid evolution in host tolerance, pathogen virulence, or both. Now, to sort of demonstrate this concept, I want to take you back to Panama and put forward this idea of how this might work. So in Panama, back in 2004, we had amphibians that occurred across this altitudinal gradient. Now, at that time, BD moved into these populations very rapidly, as I mentioned. Now, because of the thermal envelope at the higher elevations, we had these mass mortality events. Again, because BD is most effective in temperatures that match the temperature range up in the high elevations. So, presumably, there may be some survivors. They may have some sort of protection against disease development. We may also see in this case that we have a BD that may have a slightly lower level of virulence after a few generations. So fast forward from 2004 to 2014, say, 10 years later. Will we see evidence in these lower populations that suggests that there is weaker selection for host tolerance and higher pathogen virulence? Possibly. Now, up where the outbreaks occurred, where there was mass mortality, will we see strong selection for host tolerance and lower pathogen virulence? That's also entirely possible. And there also may be a lot of permutations of both of these possibilities. These are sort of the boiled down basics of what might be going on. So the question that I am trying to begin in this investigation is to understand the role of rapid evolution in host tolerance and pathogen virulence, or both. And again, this is in the early stages, but here is how I'm starting to approach this. To begin with, characterizing Chytridiomycosis epidemiology anywhere from 5 to 10 years after the epidemic state. What are the intensities of infection now? What does prevalence look like now? Is disease occurring? Is there mortality occurring today 5 to 10 years after the outbreaks. I'm also interested in evaluating the differences between pathogen virulence, host tolerance, along a spatial temporal gradient. Now, in this case, I'm a huge benefactor of a lot of researchers that have been not only collecting information, but also preserving isolates and collecting host tissues all the time that these Chytridiomycosis epidemics have been happening. So a lot of those are stored in various universities, and BZ is one of them, where we have tissues stored, which is an incredible resource to be able to move forward on this research. And lastly, I'm going to use some more of the experimental evolution techniques to try and understand what are the mechanisms of shifts in virulence that may be occurring. So I am using these two replicate systems. Although they are dramatically different in terms of their climactic region, I think that actually provides an incredible opportunity to look at how this is happening in two biologically replicated cases. So there are also a lot of historical resources available, as I mentioned, BD infection data as well as ecological data for the host species, cryopreserved BD isolates, and preserved host tissues. The other advantage is that in both of these areas we have highly susceptible host species. We have evidence for survival of at least some individuals and potentially populations. And at least in the Panama system, there's a very clear altitudinal gradient to investigate these patterns. We also have an enormous possibility, or I guess very promising possibility, that there are animals in captive breeding that are now completely naived by BD because they were removed from those areas before BD was introduced. So their offspring, or their progeny, are a huge possibility to be able to make some comparisons between BD naive animals and those that have been infected for a long time. So a couple of preliminary findings. I spent a lot of August and September up in the high elevation of the Sierra Nevada, going back to these old sites and seeing what's there. Although some of these areas, there's a lot of lakes that have absolutely no amphibians in them. There are some glimmers of hope in there. We've found at least one, potentially two, populations where the amphibians are actually doing quite well. They look healthy. There are signs of recruitment. We have individuals who are just getting to reproductive age, as well as a lot of tadpoles and small little meadowlings. So this is incredibly promising, and I look forward to being back here next summer. In terms of the Panama system, I leave today, so I'll have to let you know in a few weeks' time what we find there. There is anecdotal evidence that there are Atelopus species there. In addition to Atelopus, we will also be looking for species that we know are doing well in these BD infected areas, and we're going to be collecting a lot more information on species that at the moment we know nothing about. So more to come in terms of wrapping up this particular chapter. So with all that presented, I do not only want to give you a sense of suspense in terms of what is to come, but also a message of hope. So, unfortunately, pathogens are going to move around the world. This clip is of air traffic control, and every little yellow dot that you see there is an airplane. But those airplanes aren't just carrying humans, of course. They're also carrying animals as well as plants, and also their pathogens. So pathogens are inevitably going to be everywhere the more connected our globe becomes. This is not going unnoticed by a lot of different scientists and researchers. So in a recent meeting with human medical mycologists, wildlife biologists, and plant pathologists, we're discussing diseases that are incredibly devastating for a huge range of organisms. BD, the one that we talked about today, is actually an excellent company with a lot of pathogens that have come out, emerged, and are doing very bad things to their hosts. Not the only villain. In particular, you may be aware of white nose syndrome, which is affecting the bats of North America. It's very similar to Chytridiomycosis in a lot of respects. It's causing mass mortality events estimated at many millions of bats. It's spreading rapidly, and there is a very complex host-pathogen interaction that we're just beginning to understand. But with all that said, we are making progress. In studying these diseases for many years now, we are starting to figure out the complexities of these diseases. Now that's important, not only just to understand the basic biology of the disease, but also to implement protection measures or conservation measures. So how we view these problems is really important. And the question of how does it end, I'm going to have to come back in about 100 million years and tell you a little bit more. But what I will say for today is that I think that it's time to stop documenting declines and focus on the survivors. I think it's really important to understand why they're surviving, if we can understand the mechanisms of evolutionary shifts in these disease systems, so that we can hopefully help these populations to recover and help them to perpetuate well into the future on planet Earth. With that epilogue, I will begin with my acknowledgements. I do have a very long list of people to thank, but I just want to give a very special thank you to Bree Rosenblum, because she has been absolutely the best advisor any postdoc or student could ever ask for, and I am so grateful for everything that she has done for me. I also really appreciate the rest of the Rosenblum Lab, which is a very fun group, not only at the University of Idaho, but here at Berkeley. And unfortunately, we have newcomers to the lab that I didn't get a picture yet, but also you guys as well. And of course, the many other people who have helped me along the way. It's been a tremendous journey, and I really look forward to figuring out how this story ends. So thank you, and I'd be so happy to take any questions that you have.
This site where you found the apparent thriving population of mountain yellow-legged frogs this summer, was that a site where they were thought to have been extinct when the BD went through a few years ago?
Yes, sir.
So a few had survived, apparently hiding away somewhere.
Yes, sir. Yeah,
And there's still BD loads there?
correct.
Regarding these antimicrobial peptide profiles, how much of that is due to host production versus commensal bacteria production?
Versus commensal bacteria production?
Yes.
Well,
And
in
how
our
much intraspecific variation has been seen in these
Intraspecific variation in the peptides? Yeah, there definitely is some. A lot of that work has been done by Doug Woodhams. So I guess I should also restate the question. The question is about antimicrobial peptides and what's the intraspecific variation in them. And there is a bit, but they're very, but not as much as the species differences. So interspecific variation is dramatically different for antimicrobial peptides. And then the first part of your question was about microbes.
just the peptide production, host versus commensal bacteria?
Yeah, and that's also an excellent question. So a whole nother chapter of this story centers on these cutaneous microbiota, so the bacteria that are found on frog skin. To the extent that we can figure out what are the peptides responsible versus what's produced by the microbes, that's something that's being teased apart now by a lot of different researchers. Been an exciting chapter, for sure. Yes, Steve?
It seems to me that one of the problems with the Castro and Volker analysis was that it was focused too much on single species, whereas you've got, in the tropics in particular, you've got so many species, so that if a single species that's particularly susceptible is declining, it's got all kinds of possibilities of reinfection from just the availability of all of the other species.
Yeah.
And even up in the Sierra Nevada, you have other species of frogs
Right.
in with the. So I'm just wondering how much their study really does apply to this situation where you've got multiple hosts.
Right, so I think Dave's question, if I can restate it, is just to what extent can we use the mechanisms put forward in DeCastro and Volker to understand how extinction might happen in a community assemblage? And I think that that is a really important point, because not only do we have other species that can be reservoirs for the pathogen, but also presumably other non-amphibian organisms or even in the environment. And so, yes, I agree with you. I think that they consider single species in isolation when what's needed maybe is a more holistic approach and understanding what's happening for the community assemblage, yes. I would say that it's the first paper that's, that was put out there and said, we looked at all these cases and actually there is a bit of support for this idea that disease could cause extinction. So, but yes, I do think that it needs to be probably not only take a step back and take a bigger view and refine those mechanisms and our understanding of them.
And there is the other issue of local versus global extinctions in some of these species. For
Yes.
example, at Monteverde, a whole bunch of local populations went extinct.
Yeah.
Most of them had broader distributions.
Yeah, yes, I agree with you. I think that there's a whole lot more that we need to understand about the disease-induced extinctions and the mechanisms underpinning them. Yes, Jim.
So the El Cope site, I mean, is there any evidence of a rebound there? I mean, people must have been following what's happening to the amphibian populations there following the big sudden drop-off. Is there any good news from that site, or
I
are
can
they
tell you in 6 weeks' time.
So you're going there?
Yeah.
Ah, okay. And nobody else has been doing work there in
No,
the
there's,
interval?
Karen Lips, who has been following that system for a long time, she periodically has students who go back in there and run transects, but not in a systematic way, unfortunately. Yeah, unfortunately. I think her research focus sort of shifted to the eastern side of Panama. And I think anyone who's working in the tropics also can feel, it gets so complex that, and there's so many different things going on that you have to pick and choose your battles, I suppose. But yeah, that's the idea. And I'm going back with Corey, your former postdoc.
Oh yeah.
Yeah.
Excellent.
And so we are doing resurveys of her transects from her PhD work.
Is it her work that suggests that maybe there's been some rebound in Atelopus?
Well, actually, it's anecdotally, people have been talking about finding Atelopus. So Atelopus actually is the magic animal for Panama. It's on their lottery tickets. And so one thing that we want to be careful of is there is a lot of harvest of these species for the illegal pet trade. And so we want to be careful to not call attention to any surviving populations as well, right? So that's sort of a fine balance in terms of conservation objectives. You want to know that they're there, but you don't want too many other people to know that they're there. Not yet, anyway. Yeah. Yeah.
Jane, is there any evidence that baby fungus is evolving tolerance to higher temperatures? I know your experiments show that BD doesn't grow after 30 Celsius, but is there any place where people have found higher temperature tolerance?
Yeah, excellent question. We're testing that right now with a student from Berkeley named Rochelle Kelly, and we actually are doing that experiment right now. I didn't have the data available to crunch, but it does look like, interestingly, to my surprise, that isolates from temperate regions seem to have a higher thermal maximum tolerance, a higher thermal tolerance than the ones from tropical regions. So, so it's coming very soon.
Okay, last question. So you were showing some maps, and I was just kind of curious about your thoughts on the continuing southward spread of BD, because obviously we were lucky to have Vance kind of working on the northern end and when watching these events occur. But are there people working, or do you have thoughts on sort of the monitoring for that front that that's apparent in at least in Panama right now, it seems?
So that is something that Karen Lips has been working on. So she has sort of looked at two different fronts, one coming up from South America and then the one going down from Panama towards the canal, which is now on the other side of the canal. And so to the extent that she is able to find the front, which is not an easy thing to do, right? Like it's really amazing that it was possible in Panama, but I think it's because it's a really thin section of land where you can find that. So yes, I think that there are people who are still trying to find the front. There was a huge effort in the Sierra Nevada this summer to find populations where they thought there was going to be an outbreak and do a mitigation effort just at the exact moment when BD was invading. In terms of how well that'll be a conservation measure, I don't know. I think that it's very optimistic, but I don't want to say never. Can you catch it before it gets there and do something to help the frogs get through it? Yeah, that would be the ideal. So I think I hit on the essence of your question.
Yeah, that could be chapter 12.
Sorry?
I said that could be your last
Chapter 12, yes. Yes.
Epilogue.
Yeah.
Epilogue.
Right.
Yes.
Let's thank Jamie.
To the credit of
I didn't have the specimens.